Point Mutations by Cassandra Gray. This is a double-stranded segment of DNA. The goal is to identify the products of transcription and translation that result in a 4-amino acid protein. In order to identify which strand is the template strand, I start by looking for a codon that reads TAC in the 3' to 5' direction, since this would result in an AUG or start codon in mRNA. Once one is found, I continue down the strand until a stop codon is found. In this case, ATT would be a UAA in mRNA, which is a stop codon. There are 3 codons between the start and stop, so this segment would result in the 4-amino acid long protein we are looking for, making the top strand the template strand, the bottom strand the coding strand, and transcription going from right to left. This slide shows the coding strand in the 5' to 3' direction, as well as the resulting mRNA and protein. Single nucleic acid point mutations are when one base is substituted for another. If the base is a purine and it's switched with another purine, or if the base is a pyrimidine and it's switched with another pyrimidine, then this is known as a transition. Then a transversion is when a purine is switched to a pyrimidine, or when a pyrimidine is switched to a purine. There are 4 main types of point mutations that we'll talk about. The first is a missense mutation, which is a point mutation that results in an amino acid change in the protein. Here it can be seen that this G was mutated to an A in the coding strand, which is a transversion. Following transcription, this is the mRNA produced, and following translation, this is the protein formed. It can be seen that without the mutation, there would be a valine here, but this mutation caused the codon to change and it now codes for isoleucine. The second is a nonsense mutation, which is a point mutation that results in a codon that would normally code for amino acid being changed to a stop codon. Here it can be seen that this A was mutated to a T in the coding strand, which is a transversion. Ultimately, this changed the lysine codon to a stop codon, which resulted in a shortened protein. The silent point mutation is called such because you often can't tell it happened since the codon, even though changed, codes for the same amino acid. Here the C is mutated to a T, which is a transition. The AGC and AGU codon both code for a serine here. The last point mutation is the read-through mutation, which changes a stop codon to a codon that codes for an amino acid. This means the protein will continue to grow in length. Here this A is mutated to a T, which is a transversion. The UAA stop codon here is converted to UAU, which is no longer a stop codon, but it codes for tyrosine. As seen, the protein will continue to gain amino acids in accordance with the codons.